Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Frank Macabenta, and I'm a postdoc at the California Institute of Technology, and will be moderating today with Dr. Jennifer McKey, a postdoc at Duke University. We are excited to highlight the work of our outstanding postdoctoral members. Today, Dr. Joey Ghersi from Yale University and Dr. Elle Roberson from the University of Texas at Austin will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. It is now my pleasure to introduce Dr. Joey Ghersi, a postdoc in Dr. Stefania Nicoli's lab at Yale University, where he studies the role of microRNAs in controlling vascular and hematopoietic developmental programs using zebrafish as a model system. Joey's expertise spans the gamut of genetics, developmental, cellular, and RNA biology, and his research throughout the graduate and postdoctoral phases of his career has expanded our current understanding of how diverse processes such as signal transduction and N-glycosylation contribute to the regulatory programs governing erythropoiesis, hematopoiesis, and vasculogenesis. Today, he will be presenting his current work on the regulatory role of microRNAs in hematopoiesis. Without further ado, I present Dr. Joey Ghersi.
Hi, everyone. So my name is Joey Ghersi. First, I would like to thank you for the really kind introduction. And also thank the organizer to give me the opportunity to share my work today with you. So today I'm going to present you my project concerning this particular microRNA, miR-128, that limits the transdifferentiation potential of the endothelium to blood stem cells. Indeed, it's in many blood disorders, or for example, after high chemotherapy treatment, there are many patients that need a HSC transplantation. HSC stands for hematopoietic stem cells. Indeed, at the moment, we still suffer from major limitation of the current use of the allogenic and autogenic transplantation of hematopoietic stem cells. And it is believed that the next step is the use of induced pluripotent stem cells, iPSCs, in order to produce ex vivo those HSCs. However, we still have limitation on how we can produce really true long-term HSCs from those iPSCs ex vivo. In this scenario, we really need to understand better how those HSCs form in vivo and what is really regulating their formation. HSCs in the adult, they reside in the bone marrow. Those HSCs able to self-renew and to produce all the blood cells. We have the three main blood lineages, the lymphoid, the myeloid, and the erythroid. Importantly, the pool of HSCs that you have in the adult is only originating from a specific time window in the embryos through a specific mechanism called the endothelial to hematopoietic transition, also called EHT. Indeed, it's from an endothelial cells that some of them will acquire this hemogenic potential and then to become this hemogenic endothelial cells, HEMICs, and then differentiate into this nascent hematopoietic and progenitor stem cells, and HSPCs. This mechanism is conserved across vertebrates. And here in the lab, we are using the really wonderful model, the zebrafish model. Indeed, it's transparent and we can see in vivo, for example, this EHT happening. This is really important. What is happening, this EHT is only happening in this ventral floor of the dorsal aorta, VDA, whereas the cells, the endothelial cells in the dorsal floor of the dorsal aorta do not undergo EHT. And as you can see here in this time-lapse movie, we can see some of these red cells having this green fluorescent protein, GFP, under the control of the SEMI, which is a well-known transcription factor expressed in the nascent HS species. You can see that those endothelial cells transdifferentiate into this nascent HS species. What is happening next after they're forming? Indeed, after they're forming from 25 to around 48 hours post-fertilization in this VDA, they will migrate to the second hematopoietic tissue, the caudal hematopoietic tissue, CHT. There, from 40 hours to 6 days post-fertilization, they will self-renew, but also differentiate into erythroid and myeloid progenitors. To first see in lymphoid progenitors, we have to wait 4 days post-fertilization and then analyze their formation in the thymus specifically. And finally, some of those remaining HSCs in the CHT will finally migrate to the kidney, which is the adult hematopoietic tissue. from around 5 days post-fertilization. There they will self-renew and also form all the blood lineage cells. Okay, let's go back to the CHD. Indeed, the CHD is really, really interesting. Indeed, it requires this precise developmentally timed gene expression program, where here in red, we have this, we have to, during EHD, downregulate this endothelial gene program, while at the same time increasing the hematopoietic gene program, as you can see here in green. Since the discovery of this mechanism, multiple EHD determinants were found. Indeed, we have, for example, some transcription factor, RUNX1, CEMIB, but also some really well-known cell signaling pathways. We have, for example, Wnt beta-catenin, retinoic acid, and Notch signaling. More recently in the lab, we discovered that the N-glycosylation pathway is really important to regulate the CHD. And this pathway is regulated by this particular microRNA, miR223. However, despite this all knowledge of those different EHD determinants, we still do not we are still not able to really produce ex vivo those HSCs. So we still need to understand how those EHD determinants are regulated and how this regulation impact the HSCs formation and then their differentiation later on into all blood lineages. What I did is basically to do a phenotypic screen on CRISPR-Cas9 mutants. Indeed, not only on general mutant, but on mutant on specific endothelial enriched microRNA that we previously published. And what I found is that indeed, I found that one particular microRNA mutant, miR128 mutant, had a really interesting hematopoiesis defect. Indeed, when I did a time-lapse imaging from 26 hours to 48 hours post-fertilization from wild type and miR128 mutant, I did find that indeed we have an expansion of the EHD happening in the miR128 mutant. As you can see in wild type, we have this formation of those two nascent HS species, while in the mutant, we have more than two. We have 4 right now at the moment. So indeed, we have in the mutant an expansion of this EHD. But do we have a change in later on in the blood lineages formation? For this, as I told you, after their formation in the VDA, they will then migrate to the CHD and then there produce the erythroid and myeloid progenitors, while in the thymus produce the lymphoid progenitors. So first I analyze the number of erythroid progenitors that we have in the white type fish and in miR128 mutants. What I found is that indeed we have here, after quantification, an increase of the number of these erythroid progenitors forming in the mutant compared to the white type. Really interestingly, when I re-expressed miR128 specifically in endothelial cells, we found that we were able to rescue this increase of erythroid progenitors. And that tells us that indeed, we have this function of miR128 during EHD that affect this formation of erythroid progenitors. Then I analyzed the lymphoid progenitors in the thymus. What I found is really similar. Indeed, we have an increase of lymphoid progenitors through 1 month in situ hybridization against Icarus, which is a marker for lymphoid progenitors. So we have an increase of these lymphoid progenitors in the mutants compared to the wild type. And that increase was also rescued when we re-expressed miR128 in endothelial cells. Interestingly, we did not find any differences in the number of myeloid progenitors in the mutant. Moreover, when we re-express miR128, that do not also affect this number of myeloid progenitors. So indeed, in the mutant miR128, we have an increase and expansion of EHT that lead to an increase of specific erythroid and lymphoid progenitors, while not affecting the myeloid progenitors. I wanted to dissect a bit more the molecular phenotype happening in this miR128 mutant EHT. For this, I isolated the trunk endothelial cells from wild type and miR128 mutants, and then at the early time point of EHT, and then I performed the single cell RNA sequencing, the 10x genomic. After analyzing some gene markers, as you can see on the right, I was able to identify those EHT clusters. So cluster 2, the RTL cluster, the cluster 1, the pre-homogenic endothelial cells, homogenic endothelial cell cluster 4, and then this bigger population of nascent HS species. Indeed, this population of nascent HS species was divided into 4 clusters, where we have cluster 3 and cluster 6, so 2 clusters corresponding of multipotent nascent HS species, and 2 other clusters. At the same time, I did analyze the expression of this GATA1, Icarus, and LCP1. And I found that indeed, this cluster width cluster 8, is a lymphoid erythroid primed and nascent species through the expression of GATA1 and Icarus, while the cluster 5 is known as LMP as a lymphoid myeloid primed nascent species with the expression of LCP1 and Icarus. Now let's see if we have a change in the miR128 mutant cells compared to the wild type cells. Indeed, when I analyze the expression of GATA1, Icarus, LCP1 in the mutant cells compared to the wild type cells, I did find that we have an increase of this cluster 8 LEP, but no change of this cluster 5 LMP. And that leads us to our first model, where indeed we have this new microRNA miR128 that is able to inhibit EHT. Indeed, in the mutant, we have an expansion of EHT leading to an increase of nascent species, and that will those cells will be skewed to more to this lymphoerythroid lineages. Indeed, we have this increase of lymphoid erythroid prime nascent species. However, now the next question is, how does really miR128 is able to regulate this EHT? So for this, we performed a similar experiment that we did for the single cell experiment. We isolated the trunk endothelial cells from wild type and miR128 mutants at 26 hours post-fertilization, so early time point of EHT. And then we performed the high throughput RNA sequencing. We specifically worked on and analyzed the genes that were upregulated in the mutants, but also predicted to be direct target of miR128. And we found that indeed, a lot of genes were indeed part of the Wnt and Notch signaling, really two major EHT determinants. If you remember that my introduction, indeed Wnt and Notch are really major EHT determinants. And we focus in one gene in each of those signaling. Indeed, we focus on JAG1B, a ligand in Notch signaling, and CSNK1A1, which is part of this beta-catenin destruction complex together with GSK3 beta and actin-1, part of this Wnt signaling. Okay, so it looked like indeed miR128 is able to target Wnt and Notch during EHT. However, what does that mean? Does it really increase or decrease Wnt signaling? So for this, we went back to the single cell experiment. We first analyzed in which cells do we have an activation of Wnt and Notch. Indeed, we found that both signaling, Wnt and Notch signaling, are predominantly active prior EHT. Indeed, as you can see, we have a better activation in this cluster 2 and cluster 1, while in the cluster nascent species, we do not have any activation of both signaling. Then, interestingly, when we compared wild type cells and mutant cells, we found that indeed we have a downregulation of both signaling, specifically in this cluster 1 in pre-immunogenic endothelial cells. And that this downregulation of both signaling and the fact that this is only happening in the pre-immunogenic endothelial cells is coherent with this. Indeed, JAG1B is known as a ligand to trigger a really lower activity of Notch signaling. And CSNK1A1 is part of the destruction complex of beta-catenin. So both are known to really downregulate this activation of either Notch or Wnt. Moreover, in the single cell experiment, we found that both genes are enriched in this pre-immunogenic endothelial cell cluster. But we wanted to validate those downregulation and the implication of those genes with the different signaling pathways. So for this, we took advantage of this transgenic line. For Wnt signaling, it's going to be the TCF hemcherry. So indeed, what you're going to see that every time there is a cell that is red, so expressing hemcherry, we have activity of Wnt signaling. And then we quantified the number of cells active or not for Wnt signaling in the ventral floor of the dorsal aorta, where the EHT happening, and the dorsal floor of the dorsal aorta. We found first that in the mutant in red, we have a decrease of the activation of Wnt signaling, specifically in the ventral floor of the dorsal aorta, but not significant change in the dorsal floor of the dorsal aorta. Then what we did is to mutate the sequence of the 3' mutar of CSNK1A1, where miR128 is predicted to target. So indeed, by mutating this sequence, we will remove the capacity of miR128 to bind and do its inhibition effect on those genes, on these genes. And what we found is that indeed, when we remove the capacity of miR128 to bind CSNK1A1, we indeed also find a decrease of Wnt activity, specifically in the ventral floor of the dorsal aorta, while we do not affect the activation of Wnt signaling in the dorsal floor of the dorsal aorta. We did a similar experiment with the notch signaling. This time it's TP1-GFP transgenic line. So indeed, where notch is active, you will be able to see the expression of GFP. And after quantification, we found really similar results. We have a downregulation of notch signaling in the VDA, in the mutants, but also in the JAG1 guide RNA 3' DTR, while we do not have any change in notch signaling in the dorsal floor of the dorsal aorta. So it's telling us that it's really specific to the EHT. Then we wanted also another proof of really functional assay. Indeed, are those genes important to make this increase of HRAD or lymphoid progenitors that we found in the mutants? What we found is super interesting. Indeed, we found that CSNK1A1 contributes specifically to the HRAD lineage while not affecting the lymphoid lineage by here it's a 1 month hybridization against the GATA1 for HRAD and Icarus for the lymphoid. In the inverse, we found that indeed this miR128 JAG1B regulation do not impact these HRAD progenitors amounts, but impact and increase the lymphoid progenitors. So we found that miR128 JAG1B is required for lymphoid and miR128 CSNK1A1 required for the HRAD progenitor formation. But now, is it really the role of those genes through their signaling, so Wnt or notch, or other signalings? Indeed, what we found is let's validate that. Are really Wnt and notch important in that context of forming specifically HRAD or lymphoid? So we drug treat the embryos with super well-known inhibitors of Wnt or notch signaling during the really early time of EHT, only from the 26 hours to 32 hours post-fertilization. Then we remove the drug and we analyze the expression of GATA1 in Icarus at 4.5 days post-fertilization. And we found a similar result that indeed with the IWP, which is a drug that inhibits Wnt signaling, we have an increase of these HRAD progenitors while not affecting the lymphoid progenitors. At the inverse, when we use a DAPT drug, that is a well-known inhibitor of notch signaling, we do not affect the HRAD progenitors, but we have an increase of these lymphoid progenitors. And that led to our final model, where indeed we identified this new microRNA that is a new inhibitor of this EHT. And indeed, it's just not a general inhibitor. It specifically targets some target gene that then regulate a specific signaling during EHT. Indeed, in the miR128 mutant, where we don't have expression of miR128, we have an expansion of this nascent HSP species formation. And that happened through, first, JAG1B. Indeed, in the mutant, we have an increase of JAG1B that will lower the activity of notch signaling. But that is really important. This is specific prior EHT, specifically to these pre-hemogenic endothelial cells. And this lower activity of notch signaling prior EHT would then lead to an increase specifically of this lymphoid lineage. While when miR128 affects the CSNK1A1 Wnt signaling, that prior EHT in pre-hemogenic endothelial cells, that will lead to an increase of the HRAD lineage. So indeed, miR128 limits the EHT of prime lymphoid HRAD nascent HS species by sustaining the notch and Wnt signaling prior EHT. But that now we have new challenges, new questions. Indeed, we have to understand really better this pre-hemogenic endothelial cell heterogeneity and their priming potential. What I mean is, indeed, we found that by changing the activity of notch or Wnt or EHT determinants prior EHT in pre-hemogenic endothelial cells, that will affect the cell fate decision of those different nascent HS species. So it will be really important really important to understand from exactly, do we have different EHT, what is happening? Or are they all having a memory of what happened prior EHT and then remember later on after EHT, oh, okay, I have to go to one or the other fate. That will be really important. Next, we want to know, as I told you in the introduction, that the pool of HSC that you have in the adult is coming from this EHT. So indeed, it will be really interesting to see that if a change only in this small population of cells will have big defects later on in the adult. And I do think that this is really an important question. But now the next question is a holy grail. Indeed, we want to know if we can use this new knowledge on the regulation of EHT determinants on making new HSCs ex vivo. And for this, we started a collaboration with Christopher Sturgeon in New York. Indeed, they have a really wonderful protocol where they are using human embryonic stem cells, and they are able to recreate this EHT. And what they did, so it's done, this pilot experiment done by Stephanie Luff, a postdoc in Christopher Sturgeon Laboratory. What they did is by removing the expression of miR-128 through the non-tegomere, they were able indeed to see that in prior EHT, we have an increase of these erythroid numbers, while no change in the myeloid numbers. While when they remove activity of miR-128 after EHT, we do not affect either erythroid or myeloid. And that indeed shows us that potentially miR-128 effect on EHT is conserved across vertebrates. So there is more work to do, of course, to really understand if it's true Wnt and Notch, but this is really promising and yeah, really promising. I would like now to thanks, of course, my lab, my mentor, Stefania Nicoli. I would want to specifically thanks Jared Insen and Lily. I would like also to mention that Gabriel Baldissera did everything about the single cell experiment. So he really did a wonderful analysis that helped us in this project. I would like to thank the collaborators, so Stephanie Luff and Christopher Sturgeon, the funding source, so the American Art Association. And also I put here my email and my Twitter in case you want to contact me after the meeting. And thank you for your attention.
Thank you, Joey, for that wonderful talk. We have a question here from Wade. Wonderful talk. I'm curious, have you ever examined if these microRNAs have effects on blood formation at the level of the lateral plate mesoderm?
This is really interesting. Indeed, I did analyze some expression pattern of SCL TAL1, for example, at the early time point at 12 hours post-fertilization for the lateral plate mesoderm. By 1 month in situ hybridization or qPCR, I did not find any change in the lateral plate mesoderm. Also here, I did not have the time, but I did analyze like any vascular phenotype. And for everything I did not find any changes. Also, the quantification we did for specifically VDA and DDA that show that indeed miR-128 seems to be really specific to the EHT, so to the blood formation, in definitive way.
Thank you. I actually had a question about the cells that you showed earlier, where you showed that phenotype of the expanded EHT in the miR-128 unit. I noticed that the cells have this like aberrant migration phenotype, or it looks like it's pretty aberrant. Can you tell us more about whether there are any cell migration regulators that are potentially targeted by miR-128, and does that have any effect on the I guess the health of the embryo?
This is really a cool question. Indeed, I didn't have the time to show that, but this expansion of EHT, indeed, it's not only we have more of this nascent HSC species forming, the formation, their delamination time, so how long the cells, when there will start to be an endothelial cell, so start to be a nascent HSC species, how long the cells will stay in the VDA or leave the VDA and go to the circulation. So we were able with time-lapse imaging to quantify that time. And indeed, we found that in the mutant, those really newly formed nascent HSC species, they take a longer time to leave this ventral floors of the cell aorta. Previously also in the science paper we did last year, we found that also there is a difference in delamination time. And other people in the British also showed that sometimes when they have a change in EHT, there is a delamination time that is changing. And depending on that, you can see that some of these newly formed nascent HSC species will be more skewed to one lineage to the other. I do not know if it's a cause effect dependence or just because it's happening, there is something linked or not, but I do think that there is something to dig a bit deeper in this context, especially because we know endothelial cells react to blood flow, to mechanical forces. So it looked like really interesting and potentially a strong link between this delamination time, so delamination behavior and their health of nascent HSC species.
Thank you. And we have a question from Ashley. Is it known what regulates expression of miR-128?
Wonderful question too. Despite some trials of 1-162 hybridization, fluorescent hybridization, I was not really able, because it's a microRNA, it was really difficult to find their expression. However, what we can do, and what I'm trying to do right now, is to use a sensor. So basically, we can design a sensor where we have in this free parameter of a fluorescent protein, the sequence where miR128 will bind. And indeed, by seeing where this fluorescent protein is on or off, we can then see which cells do express miR128. So that we are trying to do. One more answer is that what we know from the previous paper is that indeed miR128 is a microRNA that is enriched in endothelial cells. But then it's true that we do not know in which exactly endothelial cells at what time point there is expression of miR128. I did a really broad qPCR expression analysis of miR128 at really like 12 hours, 24 hours, but that do not really help us on the spatial expression. Moreover, what I'm gonna do right now also is to see if there is a feedback loop of Wnt and notch signaling. Indeed, it's really well known that usually when something is regulating a signaling pathway, then there is a feedback on this regulator. So I'm trying to see if by decreasing Wnt and notch or activating Wnt and notch, I do see a change in miR128 expression. And I do think, yes, this is a really important question that we have to answer indeed. Thank you for the question.
Thank you. I guess I had one more question. Back to this movie where you showed the expanded EHT in the miR128 mutant, and I really like this model of how this single microRNA is able to affect both notch and Wnt signaling to affect the lineage. Can you differentiate which cells are actually lymphoid versus erythroid, and whether there is a tilt towards one fate versus the other in this particular mutant background?
I would love to answer yes, but indeed we can't at the moment. The only experiment that we were able to differentiate that already at this time point in this VDA, we have a change and already a prime into whatever lineages was with single cell experiments. Now the job that we have to do potentially is to dig a bit deeper in this single cell experiment and to try to find some genes that are specifically expressed in one or the other prime nascent HA species, and potentially use that to make transgenic line or I don't know, IF or antibodies or something. But it's true at the moment we don't have anything to really discriminate between a purely totipotent nascent HA species are already primed to a lineage. But that would be wonderful.
Thank you. And we have a couple of questions here. Yin Shang asked, whether, so does miR128 regulate EHT in other species, such as mouse?
Yes, good question. So indeed, there is a model of miR128 mutants. So miR128 is really well known for the brain. It's one of the most enriched microRNA in the brain. However, no one really analyzed anything for hematopoiesis in those mice. So nothing is known of miR128 affecting any type of hematopoiesis. What we found otherwise, it's with this pilot experiment, is that indeed it looked like it's not only in the breadfish, it is also happening in the human. So it would be also potentially really interesting to use a mouse model to see indeed if we have all this phenotype. But yeah, right now we are really using this model with Stephanie Love in Sturgeon Lab to see really and dig really into the molecular phenotype of miR128 effect on DCHD in human.
Thank you. And we have another question from Stephen. Maybe I missed this part, but is miR128 still expressed later in development? Mm-hmm.
It's a good question. I did not try anything really, especially in the adults yet, in terms of expression or not of miR128. What I can tell for our project is that indeed, what I did, for example, here, I will put, yes, here, indeed, I told you that this experiment that I showed you was a drug treatment, specifically of this Wnt and notch, at early time point of EHT, showing that indeed we have a change on blood progenitors, erythroidae and nephroidae. What I did also is after the EHT and try to change Wint and Nurse's drug treatment, and I couldn't find any differences. So indeed, all of what I showed today is really something that is really precise in terms of timing. Indeed, it's really specific to early EHT, prior EHT to those primogenic endothelial cells. But then again, as an next challenge is to see if there is an effect in the adult, it would be really cool to see in the differentiation of erythroid or lymphoid or myeloid even, if we have expression of miR-128 and if it affects their differentiation. So definitely something that we should continue on that.
Thank you.
Mm-hmm.
Okay, so if anyone has any further questions, please feel free to type questions in the chat or in the Q&A, and Joey will continue to answer questions through the second talk.
Thank you, Joey, for that great talk. It's my pleasure to introduce our next speaker, Dr. Elle Roberson. She received her PhD from the University of California, San Francisco, studying the mechanisms of mammalian hedgehog signaling at the primary cilium. Elle is now a postdoctoral researcher at UT Austin in the Wallingford lab, where she is investigating female reproductive tract morphogenesis using the mouse as a model organism. Elle has presented her work at numerous regional and national meetings, and the quality of her research has been recognized by several conference awards, including from the SDB, of which she is a longstanding member. She was also awarded several grants, including an NRSA F32 to support her postdoctoral research. Elle is also an alumnus of the prestigious Frontiers in Reproduction course at Woods Hole. Today, Elle will present her work on the role of hedgehog signaling during iterative modeling of the mouse uterus. And once again, if you have any questions, please remember to type them in the Q&A box, and Elle will address them at the end of her talk. The floor is yours.
Thank you so much, Jennifer, for your very kind introduction. So as Jennifer said, I'm Elle Roberson. I'm a postdoc in John Wallingford's lab at UT Austin. And I'm really excited today to tell you a little bit about an ongoing project in the lab that focuses on the role of hedgehog signaling in cyclical remodeling of the mouse uterus during the estrous cycle. So I'd like to start by just reminding those of you who don't necessarily think about the adult uterus all the time, that the adult uterus is this central organ here in the female reproductive tract, and it's the site of implantation and pregnancy. And there are two major compartments that are really important for you to remember throughout this talk. The first compartment is this inner light pink lining here. This is called the endometrium, and it's the site of implantation. The second compartment is the myometrium, which is this darker pink thick layer here that supports the endometrium. And the myometrium is composed of a very thick layer of smooth muscle that's responsible for the contractions that occur during birth. Now, there are two major drivers to my research program on the adult uterus. And the first one really focuses on wanting a better understanding of reproductive health and how it affects the person with the uterus. And I'm using reproductive health as a broad umbrella term to encompass both fertility and birth, but also uterine pathologies that aren't necessarily related to fertility. So some examples include impaired fertility, or the inability to conceive after a full year of trying. Impaired fertility affects about 13.1% of people with the uterus in the United States. We also in the US have a very high incidence of preterm birth. So about 11.5% of all live births occur preterm. And often in these instances, we don't understand the underlying reason why either impaired fertility or preterm birth is occurring. Now, as I said, there are also many uterine pathologies that aren't necessarily connected to fertility or birth, and they can arise from both the myometrium and the endometrium. And one example from the myometrium includes uterine fibroids, which are benign tumors of the smooth muscle in the myometrium, and they arise from an unknown cell type. They affect a very high percentage of the population. They also disproportionately affect people of color. And endometriosis, on the other hand, arises from a fairly unknown mechanism in the endometrium. It affects 10 to 16% of the population. And while there's no cure, this can be debilitatingly painful and also take a very long time to diagnose. So clearly, from a biomedical health perspective, there are a lot of good reasons to be studying the adult uterus in a variety of contexts. And that second major driver to my research program actually comes from my developmental biology background. And in that background, I'm very interested in how cells and tissues change their shape over time and how that change in shape and behavior impacts the tissue's function. And the adult uterus is an excellent model of this type of morphogenesis because the adult uterus undergoes cyclical remodeling every menstrual cycle. So a person can experience more than 400 menstrual cycles in their lifetime, depending on their birth control, the number of pregnancies, among other things. And we know that the menstrual cycle is driven by the cyclical secretion of steroid hormones. And I'm showing you both estrogen and progesterone, their blood concentration levels over time here. And this cyclical secretion of these steroid hormones influences changes in the endometrium. So we know that across the menstrual cycle, the endometrial thickness increases. This change in shape and behavior is essentially very important to prepare the uterus for implantation. Now, if implantation does not occur, all of that work in building up the endometrium is sloughed off during menstruation just to start all over again. Now, on average, the menstrual cycle is about 28 days long. So every 28 days in the human, the uterus is essentially undergoing scar-free regeneration. Now, it's not just thickness that's changing. That's a bit of a simplified view of this cyclical remodeling. And the way that I like to share how complex it is actually to take you back to 1908 and show you the original hand-drawn images from the original paper that first concretely shows this cyclical remodeling occurs during the menstrual cycle and isn't random. And so here you're looking at the endometrium from human samples, and on the top of both images is the uterine lumen. And you can immediately see that, yes, endometrial thickness is absolutely changing across the menstrual cycle, but it's certainly more complex. The entire architecture of the tissue is changing, including things like glandular morphogenesis. And in the century or so since, we now know that things like vascularization, secretion, and innervation are all significantly changing across the menstrual cycle, among other things. Now, something that we don't understand very well about the menstrual cycle is how this steroid hormone signaling impinges on other signaling pathways that then help control changes in endometrial remodeling. And when I started this project, I was really interested in taking more of a molecular genetic approach. And so instead of using humans as a model system, I'm using the mouse. And the mouse uterus has a very similar structure to the human uterus. This is a cartoon cross-section through the mouse uterus. You'll see data that looks very similar to this in a couple of slides. And just like the human, the mouse has a central lumen. It's lined by a luminal epithelium. Glands branch down from the luminal epithelium, and this epithelium is supported by a thick layer of fibroblasts called the stroma. And together, these cell types make up the endometrial compartment. Now, the endometrium is supported by two thick layers of smooth muscle in the compartment called the myometrium. One of the major differences between the mouse and human that's relevant for today is that the mouse doesn't actually menstruate. Instead, it undergoes what's called an estrous cycle. And that's because since the mouse doesn't menstruate, it instead resorbs that uterine lining, which is why it's called an estrous cycle. So luckily, the estrous cycle in rodents is fairly rapid and also fairly easy to track. And it's broken down into 4 different stages, the names of which don't actually matter. But what is important is that each stage takes approximately 24 hours to progress through. And so on average in the mouse, the estrous cycle is 4 to 5 days long. And every 24 hours, the mouse uterus does undergo some really interesting, significant remodeling, which you can see visualized here in these cross sections through the mouse uterus at each cycle stage. And I've demarcated the boundary between the endometrium and the myometrium with this dotted white line. So with the help of an undergraduate, Harrison Mark, and a high school student, Anushka Gadambe, we've been quantifying the changes that occur in the mouse uterus over the estrous cycle. And these include things like how the total uterine area, the lumen area, and the endometrial area are all significantly shifting over time. So in all of these instances, in the first half of the cycle, the area is quite big or thick. And in the second half, it thins or is smaller. But we can also quantify how things maybe don't change, including things like myometrial area, which remains fairly constant throughout the estrous cycle. Now, some of this remodeling had been shown a couple of times in the past, but again, what we don't understand about this remodeling, and actually the major driver of this project, is asking what these signaling pathways are that connect steroid hormone signaling to uterine remodeling during the estrous cycle. And my overarching hypothesis is that it's going to be developmental signaling that's associated with uterine remodeling. So to begin asking this question, I performed RNA-seq on whole uterus at each stage of the estrous cycle. You can see that data set visualized here in this PCA plot. In this plot, each point is one sample. The samples are color-coded based off of their cycle stage. And the way that I like to interpret a PCA plot is that the closer 2 points are to one another, the more similar they are transcriptionally. But the farther apart 2 points are from one another, the less similar they are. So you can see pretty quickly that within cycle stage, the samples are fairly similar, although this yellow met-estrous stage is a little bit different. This is either due to a failure of estrus tracking appropriately, but it could also very well be due to biological variability. But regardless, between cycle stage, you can see that the points are fairly far apart from one another. And this suggests to me that there's radical gene expression changes that are occurring in the mouse uterus every 24 hours. And while I don't have time to show you the volcano plots, what I can say is that as the uterus transitions from one stage to the next, there are hundreds of genes that are being differentially regulated. I do want to give you a sense, though, of these gene expression changes. And so to do that, I'll show you the top 15 or 20 most variably regulated genes across the estrous cycle. So in this and all the other heat maps, each column represents one sample, and the samples are clustered by cycle stage. Each row is one gene, and the gene expression is row normalized, such that high gene expression comparatively across the row is in brown, and comparatively low gene expression is in blue. And you can just see this beautiful cascade of differentially expressed genes that is occurring across the estrous cycle. Now, as I was performing the initial analysis of these differentially expressed genes, gratifyingly, as the uterus is shifting into the second half of the cycle, the majority of the go terms include development and morphogenesis. And so with the help of an undergraduate turned technician, Kim Tran, we've started to delve into what these developmental signaling pathways and developmental genes are that are differentially regulated. And here I'm just showing you some heat maps for Hox genes, TGF-beta signaling, and Wnt signaling. We've identified a couple of other signaling pathways as well that I'm not showing you. But what we thought was most exciting actually was hedgehog signaling. And that's because unlike these signaling pathways and genes, all of the core hedgehog signaling genes are enriched together at the end of the cycle. On the left here, what I'm showing you is a biochemical pathway of hedgehog signaling. And it's not actually important for you to know the biochemical pathway. It's really here to illustrate how many of these genes are being differentially regulated across the estrous cycle. So Indian hedgehog is actually a bit of a control for us because it's known that it's a direct target of progesterone in both mouse and humans in the uterus, and it's upregulated when progesterone is high. Similarly, the downstream target genes of the pathway, GLI1, PASH1, and PASH2, these are also upregulated, which again makes sense if hedgehog signaling is upregulated anyway. But what was pretty shocking to me was that the rest of the pathway is also upregulated. Now, we don't have any idea what hedgehog signaling is doing to control cyclical uterine remodeling across the estrous cycle. And to ask this question, I've made conditional knockouts of smoothen, which is this major activator of the hedgehog signaling pathway. And to conditionally knock out smoothen, I'm using a very widely used Cre line in the fertility and reproduction fields called progesterone receptor Cre. This Cre line is so widely used because it's expressed in almost every single cell type in the uterus. So I'm showing you that with this whole mount of the female reproductive tract here, where I've lineage traced the Cre line using the ROSA 26 MTMG allele. And essentially, how this allele works is that wherever the Cre is expressed, you'll see GFP expression. And so you can see in the uterus here and here that the uterus is enriched with green. So we've been performing controls to ensure that the conditional knockout is behaving as we would expect. First, by performing qPCR to show that smoothened is significantly reduced or absent in the conditional knockout, and you can see that it is. We're also looking at signaling, the hedgehog signaling, and so we're looking at the downstream target genes like GLI1 to see how they're behaving. And what I just told you is that GLI1 is upregulated at diestrus, which you can see recapitulated here. But in the absence of smoothened, we lose that increase in expression. And so this conditional knockout is behaving as we would expect at the signaling level. One of the last controls that we performed is looking at fertility. And that's because decades of work has gone into understanding the role of hedgehog signaling, specifically at implantation. And so we know that Indian hedgehog and smooth end are required for implantation, and I've recapitulated that data here in a breeding trial showing that the smooth end conditional knockout is unable to have pups. But again, what we don't understand about hedgehog signaling in the uterus is how or if hedgehog signaling controls cyclical uterine remodeling. And what we're starting to show in the lab is that it does actually seem to be quite important. So we've taken samples at estrus, where the uterus should be quite big, and at diestrus, where the uterus should be much thinner. And you can see that recapitulated in the controls here. Again, the dotted white line is showing you the boundary between the endometrium and the myometrium. Now in the smooth and conditional knockout, when the uterus should be big, instead it's much smaller. And we can quantify that in this graph here on the right. And at diestrus, when the uterus should be small, the uterus is still small, but now there is a weird myometrial phenotype. So you can see that the ratio between the endometrium here on the inside and the myometrium has shifted. And we've quantified that as well and showed that all of the other samples have a fairly constant ratio between their myometrium and their endometrium. But the conditional knockout at diestrus has a skewed ratio. So the myometrium is taking up more area. This was surprising to us because it's not been shown yet that hedgehog signaling plays a role in the uterine myometrium. And so this is what we're continuing to follow up on now. One of the phenotypes that jumped out to us at first was just how many invaginations and how ruffled the myometrium is. And one really easy way to visualize that is just to show you the outlines of these sections. And so what we're finding is that all of the other samples tend to have a very smooth outer myometrium, but in contrast, the conditional knockout at diestrus is much more ruffled. Now we're in the middle of quantifying both the number and the depth of these ruffles now. In addition to the ruffled nature of the myometrium, it's also just thicker. And so that's really easily visualized when I zoom in to the myometrium here. Now the dotted line is showing you the boundary between the outer and the inner layer of the myometrium. And I'm doing that because it's this outer layer that seems to really be changing in thickness in the conditional knockout. So you can see the other samples tend to have a consistent thickness, but in the conditional knockout at diestrus, the outer layer of the smooth muscle is much, much thicker. So ongoing work right now is trying to understand whether this change in thickness is due to a change in the number or the size of the muscle fiber. So with the help of another undergraduate, Michelle McKey, we're using another staining technique called Mason trichrome to really contrast the muscle fiber from the rest of the tissue. And so with Mason trichrome staining, the muscle is labeled red and the collagen of the extracellular matrix is labeled in blue. And you can see that each muscle fiber is encased in a layer of collagen. So we're using this contrast to take an unbiased approach and ask if the number of muscle fibers or the size of those muscle fibers change. And we're doing that by using a program called Elastic, which essentially spits out a mask of the muscle fibers. And we can then analyze those masks for both the size and the number of the fibers. And what we're preliminarily finding is that it's not the fiber number that's changing, it's actually the size of the fiber that's changing. So ongoing work in the lab is continuing to characterize this phenotype. We're asking, if this change in fiber size is due to a change in the cell number per fiber or a change in the cell size per fiber. But we're also taking a more molecular approach and looking specifically at smooth muscle markers with immunofluorescence and confocal imaging. So with that, I'd like to summarize and just remind you that I think that the cyclical remodeling that occurs across the estrus cycle is really interesting. And I've added to this literature by performing RNA-seq of the whole uterus across the estrus cycle. And among many different developmental signaling pathways, we've also shown that hedgehog signaling is all upregulated together at the end of the estrus cycle. And this hedgehog signaling seems to be very important, not only for uterine remodeling, but also for myometrial homeostasis. So in the future, when I have my own lab, I anticipate that I'll follow up on this project in a couple of different ways. First, it's completely unclear what the downstream hedgehog transcription factors are doing in the uterus. And so I'd like to identify the GLE2 and GLE3 dependent roles in cyclical uterine remodeling. It's also unknown if the myometrium is responding directly or indirectly to hedgehog signaling, and I anticipate that part of my group will focus on this question. But I'd like to take the last minute or so and tell you what I'm potentially the most excited about for the future of my lab. So I'd like to bring it back to that first slide that I told you about. I told you that preterm birth accounts for about 11.5% of all live births in the US. Now we don't understand all of the mechanisms that keep the muscle quiescent in late pregnancy. So basically, what prevents contractions before you're at full term? And I actually think that I might have a good model to start understanding this a little bit better. And so I'm going to show you, share an anecdotal and some preliminary evidence for that. So first, I'm going to bring it back to those ruffles in the myometrium. I thought this was a really interesting phenotype, and it reminds me anecdotally about what the mouse myometrium looks like just after birth. So this is a comparison to another paper's data. It's an H&E stained cross-section through the mouse uterus 1 day after birth. And you can see that the myometrium is highly ruffled, just like in my conditional knockout. Now, in addition, I have some preliminary RNA-seq of controls and smooth and conditional knockout uteri at diestrus. And what we're finding is that in the conditional knockout at diestrus, a contractility-associated gene, a gap junction called GJA1, is upregulated. And to me, this suggests that when the muscle is ruffled, it's not quiescent, because GJA1 is responsible for continuous contractions through the myometrium at birth. And so a major direction for my future lab is asking whether hedgehog signaling is part of the mechanism that regulates myometrial quiescence during late pregnancy. So with that, I'd like to thank all of you for your attention. I'd like to thank especially the organizers of this seminar series for all of their hard work to provide this opportunity. I'd also like to thank John and the rest of the Wallingford lab for all of their support, but I'd especially like to highlight the team of trainees that I've had the opportunity to work with, including the undergraduates, Harrison Mark, Michelle Nguimsap, and Kim Tran, as well as the high school student, Anushka Gadambe, who's not pictured here. I'd also like to take this opportunity to acknowledge all of my additional mentors, both at UT, but also outside of UT, along with my funding and the Frontiers in Reproduction course at MBL in Woods Hole, which really transformed the way that I think about uterine biology and reproductive health. And with that, I would love to take any questions now or later, either via email or Twitter. So thank you very much for your attention.
Thank you, Ellie. That was a beautiful talk. We have quite a few questions already in the Q&A, so I'm going to start. We have our first question from Chris Wright, who asks whether you've thrown cyclopamine into the system at any time.
That's a great question. I have not done that. I don't remember a paper that has, but what I can tell you is that in earlier papers on hedgehog signaling during implantation, people have put SAG in to try to activate the hedgehog signaling pathway even when Indian hedgehog is knocked out, and that does help recover some of the phenotype at implantation.
Great. Another question from Lauren Burns. Where does the hedgehog come from and which hedgehog is it? So that's a great question. We know a lot about hedgehog signaling at implantation. So what I'm going to tell you is what we know from implantation. The hedgehog is secreted from the luminal epithelium, and the major ligand is Indian hedgehog. All right. We have a question from Ida Chow. Are these changes seen along the entire length and in both horns of the uterus? Apologies if you mentioned this in the beginning.
I did not mention this in the beginning. Thank you, Ida, for your question. These changes are seen along the entire length of the uterus. Actually, all of the quantitation is done along the length of the uterus. We take serial sections throughout the length to average, and they're seen in both horns. The conditional knockout occurs in both horns.
Great. We have Cooper Galvin who says, awesome, thanks, Ellie. How does collecting the sample for RNA-seq work? And then I see later on parentheses, is it the same mouse for each phase?
Great question. So it is not the same mouse for each stage. What I showed on that PCA plot, each dot was one individual mouse with one individual horn. And so for each experiment, I do vaginal cytology to estrous cycle stage, and then dissect out the uterus. And then that one whole uterine horn goes for collecting RNA for RNA-seq. The other horn goes for the analysis, the remodeling analysis. I hope that answers your question.
We have a question from Nicole Edwards who asks whether hedgehog glee signaling is important for embryonic uterus development.
Yes, it is it is important for embryonic uterus development. To add to that, I imagine that maybe you're getting at the question about the Cree. The Cree does come on postnatally, still around like P10, P12.
All right. We have a question from Anil Ganja, who says, great talk. Is IAH, Indian hedgehog, mediated signaling dependent on primary cilium, and do any of the cells in the uterus have cilia?
That's a great.
While you're at it, we have a similar question from Lauren Burns, who asks whether the smooth muscle cells are ciliated.
That's a great question. I'm obsessed with primary cilia. I did my PhD on them. So, and I have looked. The preliminary answer is that lots of cells in the uterus are ciliated. In the mouse uterus, it's primarily the fibroblasts in the endometrium, so the stromal cells are almost 100% ciliated. And there are some populations of ciliated cells in the smooth muscle layer, but whether they're actually smooth muscle or fibroblasts in the smooth muscle layer, I don't know that yet. Some of our ongoing experiments right now are working to figure that out. But yes, I love where you're going with that question.
All right, we have a question from Cooper Galvin. What might be different if you did the experiments with spiny mouse, which does menstruate, and why not use them as your model?
That's also a great question. So I think that we'd find probably fairly similar changes in the gene expression across the spiny mouse's menstrual cycle in the uterus. I would be very curious how that data might relate to the human menstrual cycle. And I'm not using them as a model system right now because when I got started, they weren't really a model. That paper came out midway through my postdoc. And the lab mouse is just such an excellent genetic model system right now. And I really want to be able to do that genetic analysis. And so I think the future for this research will hopefully go towards either maybe the spiny mouse as a model, but also human endometrium organoids and stuff like that. But that's a great question. And I really hope the people who are studying the spiny mouse are going to be doing some experiments like this.
Sounds like some fun collaborations might come into play. So I have an anonymous question. Progesterone dynamics in primates, including humans, is different than in other mammals. There is no drop in progesterone concentrations in maternal and peripheral blood, which is characteristic for the mouse, for example. How do you account for this when thinking of mouse as a model for preterm birth?
Yep, I think that's a great question. So in my dreams of models, I do think that drop in progesterone in the mouse is likely part of what's controlling the role of hedgehog signaling in myometrial quiescence. What I will say is that we don't know anything about hedgehog signaling in the human at birth. So if there's an alternate mechanism for hedgehog signaling in preterm birth, I don't know. But that's a really good question and something that I'm continuing to think about.
Great. We have time for a couple more maybe. We have a question from Kara McKinley who says, first of all, hi Ellie, amazing work. And then asks, what is known about hedgehog and fibroids?
That's a great question. We know that hedgehog signaling markers are upregulated in the fibroids, but whether it's involved in the progression of them is unclear right now.
All right. So lots of questions. Question from Joshua Tyson. Is hedgehog signaling involved in the morphological changes or differences between human and mouse uteri, particularly in the length and presence of the uterine horns?
So I think maybe we can keep talking about this over email, but I think what you're getting at is like whether hedgehog signaling is involved in the development of either the single lumen or the double horn in the mouse, and I don't know. I know that hedgehog signaling is important for the development of the uterus, but I'm not sure how important it is for the two horns to form in the mouse versus the one lumen in the human.
All right. A question from Miriam Januth. I'm really sorry if I'm botching these names.
Miriam is my grad school.
Is it possible to modulate signaling at specific stages of the estrous cycle?
Right now, not that I know of. I think that I would have to use small molecules to change signaling that transiently. There's no genetic way for me to do that right now, but I would love to be able to do that in the future.
Okay. All right, that concludes our time for today. There's still more questions, and I'm sure you'll be able to address them at a later time. So I want to once again thank you, Joey and Ellie, for your excellent talks. This was a great seminar. It has been recorded and will be available on the SDB website starting on Monday. And please remember to join us for next month's seminar, which will take place on Friday, March 11th, when Alice Accorsi from the Stowers Institute and Jia-Kun Chen from the Volume Institute at Oregon Health and Science University will present. Thank you all for coming, and I hope everyone has a great weekend.